to talk to you about different method in order different method to study mRNA accumulation in different type of tissue. So we're going to see what is in situ hybridization. I don't know if you heard already about that. You can tell me maybe. Have you ever heard about these techniques, which is in situ hybridization? No? Oh, yes.
Mm.
Okay, so we're going to see that with the exercise, and I will explain you technically how it works. After, we're going to spend a little bit of time on the quantitative qPCR, so which is a very sensitive method to quantify the RNA accumulation in different tissue. And then we will talk a little bit about a genome-wide approach in order to study global gene expression. And I will take the example of this very used method, which is RNA-seq. So this is the plan for the first part of the lecture. And after, we will make a break, and we will finish these 3 hours with one last topic in molecular biology, which is the regulation of the stability of the mRNA. You may know this process as RNAi or PTGS. So did you ever heard about this, RNAi or PTGS? No, maybe, or yes. Okay, we will see later on. So, first thing that we are going to do, I'm going to share the last part of the exercise on the regulation of the growth hormone. So, can you see the exercise on your screen? I hope it works. Yes, okay. So we were starting from, we already started, and we took a lot of time because it's very complex what we did last time. The second part, that was the characterization of the transcription factor that was going to regulate specifically this gene in the anterior part of the pituitary gland. So last lecture we had, I explained you the 2 strategies in order to be able to clone the cDNA corresponding to this transcription factor. So we saw 2 different methods. One that was using the yeast hybrid system, and the second one that was using classical biochemical purification. Okay. So from that, in the second part, we have the cDNA that codes for this GHF, which is, I just remind you, the transcription factor that we managed to characterize using when we did the analysis of the promoter. So this GHF is the transcription factor that is able to recognize specifically the promoter region that we identified in the first part of the exercise, and we're going to study the expression of this transcription factor. So we will make the link between the transcriptional regulation of the GH gene, so the growth hormone gene, and the regulation of its transcription factor. So this is part number A. If you have any question, do not hesitate. So in this part number A, we are going to see 2 different methods in order to analyze the profiles of expression of this GHF. So everything is summarized in the part of the exercise that is on the screen. So on the top, you have this drawing that is a drawing of the structure of the genomic clone of the GHF. So it's a very synthesized schematic, but what is important to see here, so we have the promoter of the GHF. It's not the GH, it's GHF, the regulatory factor. The +1 start site with the little arrow, and then you see that this gene is composed by 3 exons, okay? And there is one intron here and one intron in between exon 2 and exon 1. So that's the structure of the gene, and it's to show you where, because we are going to use primer for qPCR, where the primer are able to recognize the coding sequence, but we'll talk about that later on. So the first part on the top of this figure is a semi-quantitative reverse transcription PCR using the primer number 1 and number 2 that they are specified on the top of the figure. And this semi-quantitative RT-PCR has been done starting from RNA that have been extracted from different tissue in the mice, because we're working in mice. So we've got different lanes, mRNA extracted from stomach, from liver, kidney.
There's someone waiting to be.
Sorry, because I don't see the list of the room when I'm sharing my screen. So 1 second. Why? Oh, okay, there are 2 people coming. Okay. Now I cannot have all the windows at the same time. Okay, thank you. I'll put it here. Right. So we are going to analyze the result just after. So first part of the result, it's the result coming from a semi-quantitative RT-PCR. I'm going to explain you how it works. And the second part of the figure at the bottom, figure number 8, it is, so maybe you don't understand what it is. It's white spots on a black background. This is the result in situ hybridization in rat pituitary sections. I'll explain you later on, and I have prepared some slides to show you how we can obtain this kind of results. So basically, those 2 methods are very used and classical method in order to analyze the gene expression in different tissue. So we are monitoring the quantity of RNA that is accumulated in different tissue. So you all know how art works or not? Have you ever heard about reverse transcriptase PCR? Can you tell me, because I need to know that in order to go on.
I worked on that for a little bit.
So you work on that, but does everybody in the classroom know what is RT-PCR?
I know it.
No, but do not hesitate to tell me, because it's simple.
I know it.
So, sorry?
I know it.
You know it.
Yeah, I work with it.
So the idea is it's a method that is able to quantify the quantity of a specific RNA in one sample, okay? So the first step, obviously, it's RNA extraction. So RNA is extracted in different situations, different tissue, or different time. It depends on what we are looking for. The RNA are converted by reverse transcriptase into cDNA, which is double-stranded DNA. And then we do a PCR using specific primer of the target gene in order to amplify this cDNA. And the concept of RT-PCR, it is semi-quantitative because we will see that in depth just after when we will study quantitative RT-PCR. So I come back on what I wanted to say. So the quantity of PCR product at the end is completely, more or less, it depends which phase. Okay, anyway, it's proportional to the quantity of the RNA before the reverse transcription. So it's a way to see if in different tissue a gene is expressed, and we can compare a little bit the difference in terms of level of expression. That's the concept. But so what we do, so I need to shift to the whiteboard just to explain you something. So I stop sharing this one, and I'm just going to share the other one. Okay. Okay. Is it okay? Do you see the whiteboard or not? Yes. Okay. So the idea is in red, I'm going to write the mRNA, and in black, I'm going to write the DNA, not to mix both of the type of the molecule. So at the beginning, in one tissue, we extract RNA. The messenger RNA, they contain a poly-A tail. This we already saw it. So the first step is a reverse transcription. Okay. So this is done using a reverse transcriptase, and this will convert the RNA to a first strand cDNA. So I forgot to tell you, this is 3' here, 5' here, and this is going to be 5' and 3'. Okay, the second step is the synthesis of the second strand cDNA. So I'm not going to go into detail, but at the end, what we get is a double-stranded version of DNA that is the exact copy of the mRNA. So this is double-stranded DNA. So we call that in general cDNA. Is it fine, this? And the idea is very simple, is that if we compare, so if we extract, I'm going to write it here. If we extract, so we have situation number 1, that can be one tissue, situation number 2. If we extract the RNA contained in these 2 different samples, then we're going to produce the corresponding cDNA in both of the situation. And if we do the PCR after, so we're going to do what we call RT-PCR, we are going to do a PCR on the cDNA that have been produced from the RNA extracted in the 2 samples. After, we will have PCR product. So you can have a gel like that. You load your PCR products and you will see different intensities of PCR products, or different quantities that are proportional. The difference of the intensity, the difference of the quantity between the cDNA is proportional to the difference of the quantity of RNA that was contained in the initial samples. So it's a way to monitor gene expression. There is somebody else that is coming. So this is reverse transcription RT-PCR. So RT-PCR. So I just want to come back on one thing in order for you to understand the figure that we are going to analyze. So you see that the cDNA, after, we're going to use primer in order to amplify this cDNA. And this is going to be the PCR product that we analyze on gel. So we do a PCR. And then we do a gel. Okay, so what is important to understand is that the cDNA are the perfect copy of the messenger RNA. So it means that cDNA never contain intronic sequences. That's very important. Then that's the first point. The second point is a technical point. The technical point in this kind of experiment, the first thing to do is to extract the RNA from your samples. But the problem is that all the method that we have to extract messenger RNA from a sample, you always have contamination by genomic DNA. So this contamination can be very, very low. It's not a problem, but you may have contamination of genomic DNA. So it is a problem because at the end, you extract the RNA, they are contaminated a little bit by genomic DNA. So the genomic DNA won't affect the reverse transcription process. So the cDNA will be exactly the same as the messenger RNA. That's not a problem. But at the PCR step, you will amplify two different PCR products. The first one is the one that you want to see, the PCR product coming from the cDNA amplification. And the second one, which is a contamination, will be the amplification of the genomic DNA. So I explain you just a little bit. I'm going to take a very simple example. So let's imagine that we want to monitor the expression of one gene that contain one intron. So two exons, exon 1, exon 2, and one intron. Okay. The messenger RNA will contain only the intron, the exon. The intron is removed. So the messenger RNA will be something like that. All right, with a poly A tail. And this is the result of the fusion of the two intersplicing. Okay, so the trick in order to monitor only the PCR product that are coming from the amplification of the cDNA and not from the genomic DNA, we choose the primer for the PCR, these two primers, the one on the left and the one on the right. We choose them to be on two different exons. So it means that there is an intron sequence in between. So what will be in PCR using, so I'm going to call this primer number 1 and this one number 2. If we do a PCR with the primer number 1 and number 2 on a cDNA extraction that is not contaminated. So you can have the PCR product only corresponding to the cDNA. So let's say it's okay. But if you amplify, if there is genomic DNA contamination in the sample, you will amplify a bigger PCR product because the primer will amplify between primer number 1 and primer number 2. So it means the same sequence as cDNA plus the size of the intron. So this will be the amplification of the cDNA, but the amplification of the genomic DNA will give rise to a bigger PCR product. Do you understand this idea? So it's a way to differentiate between the cDNA PCR product and the genomic DNA PCR product that is coming from a contamination, but you cannot avoid it technically. So it's a very simple thing because according to the size of the PCR product, it will be either contamination or the real amplification of the cDNA. So is it okay for everybody? So if it's okay, I'm going to shift back to the exercise and you will completely understand what was done. So I need to find back the... Okay. So you see back the figure 7? It's okay? Yes, okay. So here, this is exactly what they did. So you have exactly the same drawing that I did on the whiteboard. So you have the genomic structure of this famous GEHF, 3 exon, 2 intron, the sites of reconnection of the 2 primer used in the PCR, and you see that they have been chosen in 2 different exonic sequences that are flanking 1 introns. So they did the PCR on cDNA coming from different organs. So you see cortex, heart, muscle, testes, pituitary, kidney, liver, and stomach. And they loaded all the PCR product into this agarose gel. So you see that first analysis of this, there are two bands, clearly. One in most of the sample, but for the pituitary sample, you've got two different sizes of PCR product. So what is happening here, according to you, what is the PCR product A, so the biggest PCR product in size, according to you, and from what we just saw, where does it coming from? Do you have an idea? We don't have the size, but it's not a problem. So we have two PCR products, one which is bigger than the other one, and the configuration is this one. This PCR is done on cDNA that have been produced from this different tissue. So according to you, the big band on the top, what does it correspond to? The genomic DNA or the cDNA?
Oh, the bigger band, the A band on top is from the genomic DNA. That is,
Yeah, that's perfect.
All of those, and then only in a pituitary gland does that DNA gets transcribed into RNA.
Okay.
For the cDNA.
So it means that the band B is the PCR product coming from the cDNA, so from the RNA. Okay.
Yep.
So it means that you are completely right, Zhang. So it means that all the top bands that you see are contaminations from genomic DNA. You understand that? So the only signal that we need to analyze is the band B, because it's the band that is coming from the amplification of only the cDNA, and the cDNA are coming only from mRNA. Is it fine? Okay. So what can you draw? What kind of conclusion can you draw from here? So if I ask you the question, in which tissue the GHF, so the transcription factor, is expressed?
In the pituitary.
It's in the pituitary, okay? You have a signal only in pituitary. So does it make sense, this? Think about what we studied before. Before we were studying the gene that is coding for the growth hormone. Okay, so the gene that is producing the growth hormone, and we saw that this gene was only expressed in pituitary, okay, and was under the control of GHF transcription factor. And this first experiment tells you that the GHF factor is also expressed only in pituitary. So does it make sense? Is it logical? It makes sense. Okay. So we are going now to see another method, and I will show you how it works, because it's quite interesting. That gives us very, very good information on in which cells the one gene is expressed. So it's called in situ hybridization. Okay, so this image here, you may think that it's very bad quality, but actually it is a very good quality image doing this kind of experiment. I propose to you before we analyze this image to shift just one thing. Did you receive the PDF files from Hang this morning?
Yes.
Yes, you have them. Okay, perfect. So I'm going to shift to another part of the slide that you already received, and it's to show you how technically we do in situ hybridization, and what kind of type of information can we obtain from this experiment, and then after we will analyze this. So I'm going to share the other part of the lecture. So Okay, I put it full screen if I manage. Okay, so this is, you have a few slides which are all the techniques that we're going to see this morning about analysis of gene expression, which is mainly accumulation, mRNA accumulation and quantification. So just a quick explanation about what is in situ hybridization. And you will see it's very nice, it's very powerful. It is not quantitative at all. So the idea, so the type of result that you have, so I want to show you a better explanation. So what you see here on the left, this is in situ hybridization done in Drosophila eggs. And on the right, it's on cross-section of a plant stem, a monocot plant stem. But anyway, the idea of in situ hybridization is a mix of histochemical analysis, so microscopic analysis, and molecular biology. The idea is if we take one tissue and we can make cross-section of this tissue, so like you can see here, it's the cross-section of a stem of a plant. These are cross-section of Drosophila eggs. We can directly hybridize with a probe. We can directly detect in the slice, in the histochemical slice, which cells are accumulating one specific RNA. Do you understand the idea? Okay, so you are just using, so technically it is very, very tricky to do because it's very, very complicated. It's very time consuming, but the results are very, very interesting because you have the profile of expression of one gene, cell by cell and tissue by tissue. The only drawback of this method, it is that it is not quantitative, but it's not a problem because we've got other methods that are quantitative. This one is not quantitative. But so, for example, here in that kind of in situ hybridization experiment, the signal here is observed because of the use of an antibody that is linked to an enzyme which is called phosphatase alkaline, and the presence of the RNA is shown by this purple color. So you see here you've got magnification of this. So for example, this small square here is the magnification here, and you can see that there are some cells, the ones that are the more purple, that are specifically expressing this gene and not the others. It is the same it's very nice for all the work that has been done, for example, in Drosophila egg, if you look on the left. So this is, you have different number SL5, SL6, so it's different stage of development of the egg. It's not important to understand. And they did cross-section of the eggs of Drosophila. They hybridize with a probe specific that can specifically recognize one RNA that was studied. This RNA is called HuncBAC in that case. So it's not important to know. So what you see here, all the dark purple signal, and you see the signal is changing over time during the development. Anyway, the purple signal, so it is an egg. So one egg is one cell, okay? So here we are looking the localization of the mRNA of HuncBAC gene inside the egg of the Drosophila. And you see that, for example, at the very early stage, the mRNA is localized only on one side of the egg. Then after, you have some kind of stripe profile that is appearing, and so on and so on. And this is what will define the head from the tail of the Drosophila, little by little. So do you understand the strategy of this technique? So the idea, you take cross-section or wall mount tissue, and you directly hybridize inside the section or inside the tissue with a specific probe, and so you can just have you can have access to which cells are expressing the gene. Is it okay, this idea? You understand the process? So all the signals in all the picture that you see now correspond to the cells that are over accumulating the RNA that is corresponding to the probe. It's fine or not? Did you understand the idea of this technique? Yes, it's okay. So I just add one more slide, which is very technical, but it is important as well for this in situ hybridization experiment. Because you have to understand that this method is very, very complicated to set up because there is a lot of background signal all the time. The idea is you take a section of a tissue and you will hybridize with the complementary probe of the mRNA target. Okay, so that's fine. This is, you are supposed to target only the specific mRNA. This is conceptually, it's okay. But in practice, you have a lot of background noise. So the controls in this type of experiment, in situ hybridization, are very, very important. And I just put this slide because if once in your research you are going to do in situ hybridization, you will need to do this. Actually, the control in in situ hybridization are almost as important as the result. Okay, so the idea is you want to use a specific probe to detect cell by cell where the gene is active, where the mRNA is present. And because there is a lot of non-specific binding of the probe, we need to use another probe which is different from the target and do the experiment on almost the same sections in order to have an idea of the general background of the hybridization. Do you understand this idea of the negative control? So it means that all the time we do an hybridization with the specific probe, we need to do the same hybridization but with a non-specific probe in order to evaluate the global background of the experiment. And so there is a trick in order to do that, is that we do what we call the sense probe and the antisense probe. So this is what is explained a little bit in this. So it's a summary of the strategy. So the principle is we want to make two different probes, one that is going to specifically recognize the messenger RNA that we want to monitor, and the other one that will be completely non-specific. So this will be our negative control. And there is a trick in order to have the probe and the control probe is to use the same cDNA, so which is the target gene, and we can make probe using in vitro transcription. Maybe you never saw that, but it's not a problem. But technically, it means that we can make probe that are called antisense probe, and this probe will match perfectly with the messenger RNA because they are antisense to the messenger RNA. So these probes will give you the signal of expression, and the control probes, to have an idea of the background, are called sense probe. And sense probe, basically, they are probes, RNA probes, that are exactly the same as the mRNA present in the cells. So because they are identical, they will never be able to hybridize together. Okay? And this probe we can detect them because when we are producing them, we are adding little flag to the nucleotide that can be specifically recognized by an antibody. So that's the way we immunologically detect the probes. Okay. So do you understand the concept? So in situ hybridization is the capacity of localization of one specific mRNA inside cross-section. But because there is an extremely high background in such type of experiment, we need to pay a lot of attention to the control experiment. And in order to have good control experiment, what we do is we make one probe that is going to be specific to our target and one probe that is going to be exactly the same sequence as the target, so it will never hybridize and you will have the level of the background of the experiment. Is it okay? It's a very short summary, but I think that you may be able to understand the idea there. Is it fine? Or not? It's okay. So I'm going to shift back to the end of the exercise where we are going to look at in situ hybridization. No, it's not this one, sorry. Okay, this one. Okay. So you can see the mouse, yes? Okay. So this figure number 8 is the result of an in situ hybridization, and this is only the picture for the positive control, so not the negative control. So you cannot imagine it, but here, if you follow my mouse, this is the form, the shape of the pituitary gland. And there is a A on the left written and a P on the right. A means anterior. Okay, so this is the front side of the gland. The gland is like that. This is the front side of the gland, and P stands for posterior, which is the back of the gland, at the back of your body. So they did cross-section of the pituitary gland, and then they hybridize with a probe that can specifically hybridize to the GHF-M messenger RNA. And the signal in that kind of experiment is the white spots. So no signal is the black background, and the signal corresponds to the front spots. So if you want to make the conclusion of the result obtained in figure set plus the result obtained in figure 8, what would be your conclusion in one sentence? What would you say when you see, if you want to analyze these two results? Can somebody of you try to tell me if it's possible? If we were all in the same classroom, we could discuss, but I know it's more complicated when using Zoom. But could you make a kind of conclusion of the result that we have obtained in these two pictures, two figures?
Yes.
The 7 and the...
Can I?
Yes, please, because it's difficult to speak using Zoom.
Yes. So
the shortest conclusion,
yes.
Okay.
From these two figures, I can come to the conclusion that first, the GHF gene is only transcribed in the pituitary gland.
Okay, perfect.
And from the figure 8, I can deduce that the expression of this gene is located on the anterior part of this gland.
Exactly. So it's like, no, you're perfectly right. So first figure, the GHF is specifically expressed in the organ pituitary, And second, you go to a higher resolution and you say inside the pituitary, it is expressed in the anterior part of the pituitary. So you see, it's like we are getting closer. So it's okay, it's perfect. Everybody understood this conclusion? So first, we target the organ. It's expressed in pituitary, and in the pituitary, it's only expressed in the anterior part. And this could have been done only with in situ hybridization. Is it fine or not? Is it okay? So just to finish, because after we go back to the mRNA analysis, just to finish this exercise, and we'll make a little drawing. So there is a case study. This is part B, okay? And it's just a study of one case of dwarfism in rats. So basically, we have normal rat and dwarfed rats. And the idea is to understand, using only this 6 result of in situ hybridization, is to understand why these dwarfed rats are dwarfed. Okay, so I come back. I just share the whiteboard to explain you one thing. Where is it? Okay. So just the big conclusion of the exercise. So first we have the GH gene, okay, and we study in part 1 the promoter. No, okay, sorry, I'm not going to draw. So first part, we study the GH gene and we study the regulatory element, and we found, so this is producing growth. So this is the gene, GH gene, and this is growth hormone. Okay. We analyze, we found the regulatory sequence, and all the electromobility shift assay, footprint, and we managed to identify the factor GHF that is able to activate transcription. From this factor is coming from the transcription and the translation of one gene, which is the GHF gene. The GHF gene is transcribed in mRNA for GF and is going to produce the GHF factor. Okay? Last thing that we didn't study is this GHF is also under the control of another gene factor. Okay, so just to make you to explain you the concept, the gene GH produce growth hormone. So if this gene is functional, the rat will be normal. So no dwarfism. Okay, no problem. This gene is only expressed in the anterior part of pituitary. Perfect. We analyze all the promoter, we isolate GHF, we study using RT-PCR and in situ hybridization, we study the localization of the messenger RNA of GHF and found that it was also, like GH, expressed in the anterior part of pituitary. Okay? So it means that this GHF is under the control of another transcription factor, once more on the top, that is giving tissue specificity to this gene. Okay, so let's come back to our rats that are either normal or dwarf. If a rat is dwarf, is because it doesn't produce the growth hormone. Okay? So there are two possibilities why it doesn't produce growth hormone. First thing is that so can you tell me at least two different possibilities that could explain why GH gene cannot be expressed? Do you have an idea?
Maybe the G. Why the GH?
Yeah, we talk about GH, and
Yes.
so if GH is not expressed, the rat, they are dwarfed. Okay.
Yes.
So what can be the two reasons? There are two reasons why GH cannot be expressed.
Either either it's because the GHF gene isn't working, it isn't transcribed, and therefore there's no binding of GHF into the promoter of GH gene.
Yes, yes.
Or another situation I could think of is the GHF is produced, that protein is produced, but it could not bind to the promoter of GH gene, and therefore no growth hormone is produced.
Okay. Okay, that's okay. That's perfect. So if I go back to your first proposition, you say GH cannot work because GHF is not working, is not transcribed. Is that the case? So this was your first possibility.
Yeah.
So if GHF in that it's completely true, that's okay, it's all right. But if GHF is not transcribed, it means that the mutation in this dwarfed rat is coming from the promoter of GHF. Because if this gene cannot be transcribed, it's coming, the problem is coming from the promoter of GHF. Okay. After you propose the second possibility, which is totally good, no problem. You are saying that the GHF gene is working. Okay, so it is transcribed. So it means no problem in the promoter. The mRNA is transcribed. The protein is produced, you say, but it cannot recognize the promoter of GH. Is that right, what you said? So in that case, you are completely right, but there are two potential origin of this problem. The first problem can come from if there is, I put it in green, if there is mutation in the protein sequence of GHF. It means that you will have a GHF that is produced that maybe is not functional. Okay? So the mutation can be in the coding sequence of GHF. The consequence is GHF is messenger is expressed, but there is a mutation in the sequence. So the protein translated GHF is not functional anymore. So it means that the mutation can come from the mutation of GHF. Do you agree with this or not? Yes. But there is a third possibility that I'm going to write in blue, is that GHF is completely normal, the protein is completely normal, functional, no problem, but the mutation is coming from the sequence of the promoter of GH. So the GHF is functional, is expressed, the protein is correct, perfect, but the mutation is on the site of recognition. So even though this protein is perfectly functional, it will never be able to bind to this part of the promoter, and therefore GH won't be expressed. Is that fine also? Okay. Yes or no? So these are the two or three possibilities. It's okay. So now we are going just to come back on the last picture of the exercise, and I explain you what we are going to look. So in the exercise, the 6 picture, they are in situ hybridization with on 2 different type of rats, normal rats and dwarfed rats, okay? And there is one in situ hybridization with GHF as a probe, and the other one is GH as a probe. So we're going to be able to see when GHF is expressed, if GH is expressed also. Okay, so I shift back to the pictures of the in situ hybridization. So I explain you how to read that. On the left part of the picture, this is cross-section of pituitary gland from normal rats, wild type. So they've got the normal size, okay? The first row is the result with a probe that is targeting GH messenger, so directly the mRNA of the GH gene. The second row is the in situ hybridization on similar section, but using GHF as a probe. Okay, so this is the site of expression of the GHF gene. Okay, and the last row, alpha GSU, is a positive control of the experiment. We're not going to talk about that. Okay, we are going to look only the 4 first pictures. So in all the situation, we have DW stand for dwarfed. So this picture, the right column, are the same cross section, but from a rat that is genetically dwarfed. Okay. P and A, it's always the same. It's posterior and anterior pituitary. So they put you the sites, and you have to look a little bit at that. So the question here is, can you make a conclusion on the molecular origin of the dwarfism in this type of rat? In other words, these rats on the right, they are dwarfed, and we want to know where the mutation is coming from, where the mutation is. Is it in GH or in GHF? The problem. So we're going to start analyzing just the mRNA accumulation in wild type rats. So you see, this is the same result as before. The gene GH is expressed in the entire part of the pituitary. Okay, is it fine, that or not? We see back the results. So normal rat, GH in anterior pituitary. We look at in this normal wild type rat where the GHF is produced. So first we see that there is a signal of GHF. So it means that GHF in the wild type is functional, and moreover, it's expressed in the anterior part of pituitary, which makes sense because this is the factor that is activating GH. Okay, so same territories of expression. Now we do the same experiment, but on rats that are dwarfed. And what do you see? You don't see. So first result, this rat does not produce anymore the growth hormone mRNA. Do you see it? Okay, so this means that the rat, they are dwarfed because they lack of growth hormone. Okay, so this is the reason why they are dwarfed. And the question is why they don't produce the growth hormone. So do you have an answer to this question? In other words, is it because their GH gene is not functional anymore, or is it coming from upstream events?
There is no growth hormone because the growth hormone factor, the GHF, isn't functional, isn't transcribed.
Alors, it is not transcribed, exactly. We cannot say if it's not functional. But the GHF, like you said, is not transcribed. So these rats, they are dwarfed not because of a problem in the GH gene, but because of a genetic problem in the GHF gene. You see? Do you understand the conclusion? And we see that the growth hormone is not expressed, the messenger are not expressed because the messenger, the mRNA of the transcription factor that is activating GH are not present as well. Okay? So this is a way to understand where the problem in the regulation process is coming from. So the regulation process, the problem in the regulation process of growth hormone production is coming from something which is upstream the GHF regulation. Is it fine? Or okay? So you see, I wanted to show you this exercise because we can see plenty of different technique and how to think about the results from that. So, okay, do you have any question on all what we see with this exercise? Otherwise, we will go on looking at two different other techniques for studying gene expression. So if you have any question on all what we saw, is it fine?
It's okay.
It's okay. So we're going to stay on the same file, the one which is called RNA analysis, where I showed you the in situ hybridization. And I want to talk to you because you will need to know that. The quantitative PCR. So do you know, have you ever heard about qPCR? It's okay. And so, I will explain you a little bit because it's very commonly used. It's very similar to the RT-PCR that we saw in the exercise, except that it's exactly the same process from a molecular biology point of view, except that it can give us quantification data very more solid than the semi-quantitative. So I just collect a few concepts that are very important in order to understand the quantitative PCR. So it's very often named qPCR. And this is the process. So I just put that again so you can keep it in mind. The same process as the the RT-PCR we saw before. We need to extract RNA from the samples. The objective of a qPCR analysis is to compare the level of expression quantitatively in different samples that can come from different tissue, from kinetics experiments, and so on and so on. So the starting point will always be RNA extraction. Second step will always be the conversion of RNA to cDNA. This is the base to every single analysis of RNA. And then we do a PCR. A PCR exactly in the same situation as normal RT-PCR. But so I just put back again the strategy of a PCR. This, you know it for a very long time. and I just want to give you a little bit of numbers and kind of calculation. So you know that the PCR cycle is doubling the number of molecule at every cycle, because the process is from one molecule. We go through different steps: denaturation, annealing of the primer, extension. So from one molecule, you end up with two molecules. So if you make a modelization of the increase of the copy number during PCR cycles, if we call X0 the number of copy at the beginning, this is very important. So how many copy we have at the beginning of the experiment, we do the PCR, and Xn will correspond to the number of copy that we managed to amplify after a certain number of PCR cycle. Okay, so you understand the signification of XO and Xn. Number of copy at the beginning and number of copy at the end of the amplification. One very important concept in quantitative PCR is that we want to quantify the quantity of RNA in a sample in order to be able to compare it with another situation. Okay, because when we do the cDNA synthesis, every single molecule of RNA will be converted to cDNA. So, second step, the PCR is amplifying the cDNA as targets. So, it means that using the PCR, if we know from how many copy we start, we will know how many copy will end. Okay, and therefore we can quantify the quantity, the initial quantity of cDNA, and therefore we can have an idea of the quantity of RNA at the beginning of the experiment. So you understand, it's a classical process: RNA extraction, cDNA synthesis. So there is a direct proportionality between the number of RNA molecules and the number of cDNA molecules, which is identical. Then the PCR to amplify the signal. And we know the relation between the initial number of the target and the final number of the target. And it's directed by this type of equation. So it's a simple equation, don't worry. Is that the final number is equal to the initial number multiplied by this amplification efficiency. I will explain you what it is, power n. N is the number of cycles of the PCR. The amplification efficiency, AE, don't pay too much attention on that, at least theoretically, because we double the amount of molecule, the numbers of molecules from the beginning to the end in one cycle. The amplification efficiency theoretical is 2. So this is the formula that explains the number of final copy in a PCR reaction related to the initial number of molecules in this PCR reaction. Is it okay, this or not? We need, they are very, very simple mathematics. And all the principle in quantitative RT-PCR is the equality of the number of molecules of RNA and cDNA and the direct proportionality between the final number of PCR product relative to the initial number of cDNA. Because if you think really well, X0 is the number of cDNA target. Okay. So starting from that, I show you the concept of qPCR. So a qPCR is a machine you don't load a gel or whatever. It is, I will explain you how the machine is reading every cycles. And this is the typical profile that we have of amplification of a cDNA in this type of qPCR machine. So it's quite simple to read. On the x-axis, it's the number of cycles. So we start with 0 cycle, 2 cycle, 3, 4, 5, 6, and so on and so on. Here, if we see, we are around 40 cycle, and it ends up at 50 cycle. It's a lot cycle. Usually we work around a few more, okay? So this is the number of cycle of the PCR. And here, it is the relative number of the product, the PCR product. Okay, so first of all, we don't see anything for quite important amount of cycle, even though, so this is the part number 1 of the curve. There is exponential amplification, but we cannot detect the PCR product yet, because I will explain you just after. You see this black line here. It's a line that is technically the threshold of detection of the machine. In other words, it means that all the value that are below this threshold line, the machine cannot really make the measurement of this. So even though there is amplification, there exponential amplification, we don't see it because it's underneath the threshold of the machine. So we don't pay attention to this. But starting from one point that we call CT, it's an important point. So CT, starting from that, it is the number of the cycle from which the quantity of PCR product goes just above the threshold. Okay, then we've got the phase number 2, which is an exponential phase of increased quantity of the PCR product. Then it settled down, and at the end, we arrive to a saturation of the 6. So in qPCR, I show you that. So this is the evolution of the number of PCR product in a PCR reaction. In qPCR, we want to work in the phase number 2, the exponential amplification, because it is during that phase that we can be sure that the mathematical relation I showed you before makes sense, okay? And it is in that phase that AE is equal to 2 in theory, okay? So all the experiment in qPCR, what you have to keep in mind, they are comparing the data coming from the exponential amplification. Okay? Do you have any question on that or not? Did you understand everything? So this phase is very important. Second other point, which is important. So I talk a lot on that, but you will understand. This so-called CT point. So it is a number of cycles. So it can be the cycle number 10, number 11, or whatever. It is the number of the cycle at which the quantity of the PCR product is getting over a defined threshold. Okay. But before, what's happening, what I was telling you before, during phase number 1, it is also exponential, but we don't see. So it means that phase number 1 and phase number 2 are exponential. It is the same. Okay, exactly the same. And what makes the difference between what we call phase 2 and phase 1 is only the possibility of seeing the PCR product or the impossibility of seeing the product because of this threshold. But this point that defines the borderline between phase 1 and phase 2, so this number of cycles, is very, very informative because it can be taken as a number that is proportional to the initial number of the molecule of X0. I explain myself. here in this graph, we've got 5 curves like I show you. So it is you see intensity of fluorescence. Don't take, we'll talk about that after. It's quantity of PCR product, okay? Quantity of PCR product in 5 different reactions. So there is the green reaction, orange, blue, red, and so on, okay? So they are on the same graph in order to be able to make the comparison. The line at the bottom here, it is the threshold line. So intrinsic threshold due to the machine. Okay, so it's technical threshold. So there is the threshold here, and you see the exponential phase and then the plateau at the end. Okay. In these 5 experiments, the numbers at the bottom correspond to the CT. So the cycle, the number of the cycle for the threshold for every of these 5 experiments. Okay. Do you follow the graph? It's okay. Then okay. And the only difference between these 5 different quantitative PCR experiment is that the number of initial molecules, so the starting template, is different. And it's for the more concentrated to the less concentrated. So it means that the green PCR reaction contains at the beginning much more matrix than the orange one, than the orange one to the blue, and so on. So we diminished the quantity of initial DNA. And what is happening is that it doesn't make completely sense, is that the number of the cycle at which the level of the signal will be above the threshold will be bigger. So the number of cycle increase when the DNA, the initial DNA decrease, because you have less molecule at the beginning, so you will need much more cycle in order to go above the threshold. Does it make sense for you? So this is what is written at the bottom. The higher the amount of initial molecule of DNA is present, the lower the value of CT. So CT equals 15 for the green reaction, which contain the more DNA at the beginning. Is it fine? And this is what I told you for A equal to 1 CT of difference correspond of a factor 2 in the amount of DNA. But okay, this is quite obvious. Did you understand well this this slide? So the message to keep in mind is that the CT value, so it means the value that are just above a fixed threshold, the CT value can give us a lot of information about the initial content of the DNA. And in qPCR, the DNA, starting material DNA, is cDNA, so it's an information about the quantity of RNA inside the samples at the beginning. Is this okay, or do you want me to come back on the explanation? So what do we do with this? Okay, so we go on. So from now, we can have, sorry, I forgot to tell you. So we've got two values now. We've got values of CT and value of quantity of template at the beginning. And if you look at mathematically, we can modelize this as a line. This is simply the plot of, if you look on the y-axis where there is my mouse, it is the CT, the cycle threshold, okay? So if you plot the CT value in y with the log of the copy number at the beginning, it's the log of x0, you have a straight line. So there is a linear correlation between the CT, so the cycle threshold, and the log of the initial copy number. And this is the most important part of all the analysis of quantitative PCR. And because the initial copy number of the DNA is proportional directly to the amount of messenger RNA at the beginning, we can directly quantify the amount of RNA in one specific sample. You understand or not? So this is what I wanted to talk to you about quantitative PCR. So it's just an idea. We can spend hours because there are plenty of different techniques. Just about quantification. Just checking the time. Okay. Quantification can always be done in two ways. There is the absolute quantification and the relative quantification. Would you be able to give me the difference between absolute quantification and relative quantification? Yes, Jiang, tell me.
I'm gonna take a guess, but
Yes, go.
I think that if we try to perform absolute quantification, we can, for example, for a certain sample, we run qPCR for it, and then we obtain the CT results. And then we will compare the CT result of that sample with a standard curve like this, a standard line like this.
Yes.
Yes, and then for the relative quantification, I think
just before the relative, so that's okay, but you compare to standard, but at the end, the value that you have, it's absolute, so it should be what kind of unit it should be?
For the CT or for the quantity?
Yeah, for the unit. When we do absolute quantification, we have a number which is the copy number, the initial copy number. So either it's a number of copy, or it's a quantity, microgram per microliter or whatever. It's a quantity, it's absolute. And this is why, like you say, you need to use standard. So it means, for example, we take one sample, we perform the qPCR experiment, we've got one result, but we don't know what is the unit of this result. It's a number. But after, if we use standard would mean we do this same qPCR experiment with known quantity of initial, of starting material. And after, we can make the correlation. So at the end, absolute quantification, you should be able to give the quantity in unit, like picograms per whatever. Okay, it's an absolute quantity. Whereas relative quantification, go on, Jiang, on what you were going to say.
Yes, so in contrast, relative quantification, maybe we can compare the CT value of two samples, and we can see that which sample has lower CT, therefore higher amount of starting template of DNA. Exactly, and the result, you express them as a fold of change. So, for example, you can say in one sample there is twofold more RNA than in the other sample, but you don't have units giving you the quantity in microgram or picograms or whatever. Okay. Is that okay? But you understand, so either you have standard for quantification, and then you can precisely quantify the quantity, the copy number of one particular mRNA, but what is done most of the time, it's relative quantification. Because, for example, you can check, you are working on one gene, and you want to know the relative expression of this gene in two different organs, brain or pituitary, whatever, and you can express that as fold of difference of one situation compared to the other. So this is the difference. But anyway, so did you
understand well the strategy of qPCR. So just to tell you how this works technically, I just put 2 slides to show you what are the chemical basis of real-time monitoring, just because there are 2 strategies, 2 big differences strategy. So there are 2 generations of qPCR machine. It's not 2 generations, it's 2 different technology. The goal is exactly the same, is real-time monitoring of PCR product. And they are both using properties of light and fluorescence. So maybe you already heard about CyberGreen. Okay, so this is one of the mostly used because it's very okay. We can read the pros and the cons. It's very cheap. It's very easy to set up, but okay, there are some cons anyway. The principle, I explain you how it works, is CyberGreen is an artificial chromophore that can give you fluorescence, but the fluorescence of that molecule is 20 times higher when it's bound to DNA. So we have low fluorescence when it's single-stranded DNA, and very strong fluorescence by binding to double-stranded DNA. So the qPCR machine that uses CyberGreen, basically it's a PCR machine like all the other one, but there is a laser light that is at the end of every cycle quantifying the fluorescence that is, and this fluorescence is directly proportional to the amount of CyberGreen bound to double-stranded DNA. So it's directly proportional to the quantity of DNA. And it's the way to monitor real-time the PCR product accumulation. Do you understand the concept? So the measurement is done at the end of every cycle. You've got a value of fluorescence, and then you plot the value of fluorescence. This is what gives you the curves that we saw just before. It's okay? So the pros of this method, very cheap, very easy to set up. The only thing that we need to do is to have a primer that are specific to the gene that we want to study. So that's very, very easy. The drawbacks of this method is that we cannot do multiplexing. Have you ever heard about multiplexing analysis?
No.
But multiplexing means in one reaction monitoring 2 different targets, at least 2, 3 or 4 different targets, not one single target. The problem with this method is we've got only one fluorochrome. So it means only one type of fluorescence. And because we have only one type of fluorescence, we can monitor only one PCR amplification at a time. So multiplexing is impossible with this CyberGreen technology. Another cons, some people say that it is a little bit less sensitive to other strategy, but it's very, very sensitive, don't worry. And there is a little drawback, which is there can be non-specific amplification. Okay, I'll show you what is a melting curve after that. The other way, so first way, CyberGreen approach. Second way, it's what is called Tachman probes. Tachman probes, it's also another strategy to monitor real-time the PCR product accumulation in the reaction. It's based on FRET. You heard about FRET concept? It means fluorescence, resonance, energy transfer. But anyway, you will hear that in other teaching unit. I explain you very quickly how it works anyway. So it's based on this principle and the fact that the Taq polymerase, which is the enzyme used for the PCR reaction, has a very strong 5' to 3' exonuclease activity. So technically, how it works? We always do a PCR. Always. So it's a classical PCR. There is a forward primer, a reverse primer that are going to amplify all the sequence between the two primers. And the Tachman probe is a third piece of single-stranded DNA, so it's almost like a primer that is homologous to the sequence in between the two primers that have been chosen. And this probe is covalently linked to two different fluorochrome. One that is a proper fluorochrome, which means that if we excite it with a specific light, it will give fluorescence. But the other one that is very close to the first one is called the quencher. And the quencher is going to absorb the fluorescence that is produced by the R part of the fluorochrome. Okay, so it means that if this molecule, this very short piece of DNA, is not degraded, it means that if you put light on R, it will emit fluorescence, but this fluorescence will be absorbed by the quencher. So the consequence is there is no fluorescence. When the DNA polymerase, the Taq polymerase, is polymerizing, so it starts from the nucleotide, from the primer, it goes 5' to 3', then it will arrive to the place where the Tachman probe is bound to the target, and because of its 5', 3' exonuclease activity, it will start to degrade the target. Because of this degradation, the first fluorochrome is going to be separate and put away from the quencher. And because this fluorochrome is not close anymore to the quencher, if you apply light, there will be fluorescence. Do you understand the principle? So the idea is that at that time, we monitor fluorescence during extension phase, okay? The advantage, okay, it's, I don't know why I put cheap, because it's more expensive, sorry, correct me because I did a mistake. It's more expensive than the Cyber one. It's very easy to set up, okay. No, sorry, I did a bad copy and paste. So I will, excuse me, because I'm just realizing that, okay, remove this because it's not I tell you what are the pros and the cons. I did a bad copy and paste of the previous one. So the pro is that it is very, very specific, because the specificity, sorry, I put it back again. The specificity of the signal is coming from the specificity of the two primers. So that's already very big. And then there is the specificity of this Tachman probe that is increasing the specificity. So that's very good. The sensitivity is higher than CyberGreen. And you can do multiplexing with that. So how can you imagine? So multiplexing, I tell you again, is in the same reaction tube, you can analyze the real-time formation of two different types of PCR product. So the idea, for example, if you want to follow the expression of two different genes, you can have two different set of primer that are targeting the two different genes, and two different Tachman probe, one for every specific gene, okay? For the first gene, the fluorochrome here and the quencher can be chosen among a wide range of different commercially available quenchers. So you can say for the first gene, you will use a Tachman probe that can be excited with red light, for example, and the emission will be in blue and so on. So we can have different fluorescent properties for different Tachman probe. And at the end, it means that within a single tube, you can monitor real-time the amplification of one PCR product and at the same time the amplification of the other PCR product. Is it okay? It's a very short summary of multiplexing, but you understand that because we have the choice for the fluorescence, we can monitor different targets, whereas using the CyberGreen, it's only one molecule that is going to bind to DNA, whatever the target is. So it's not specific at all, and this means you cannot do multiplexing with CyberGreen. Is it fine for everybody, this aspect? So it was an overview of qPCR, but at least you know the global strategy of qPCR. I hope you remember that there is a direct proportionality between the initial template and the CT at the end. So this is what makes it quantitative. Okay, and this is very and after there are technical difference depending on what type of probe we use and so on. So this is the qPCR technique. So if you have any question on this, we can talk about that now, or we will just end up, and after we make a break, just end up with a genome-wide analysis, mRNA expression analysis approach. And it's also just to give you an overview of what is RNA-seq and what can be done with this. So you have any question on the qPCR, or is it fine? Okay. So, no, sorry. There is one more slide. I completely forgot. So, this is, yes, sorry. I forgot to tell you about that. It's, okay. I need to go back just to the CyberGreen experiment. Okay. What I told you is that we cannot do multiplexing because this CyberGreen binds to double-stranded DNA, whatever the DNA is in terms of sequence, and very useful for following the real-time monitoring of PCR product. But if you have some kind of in the reaction, if you have non-specific amplification that is happening, that is very common in PCR, it's very difficult to detect because you only monitor the quantity of DNA. So there is at the end of every qPCR experiment, when done with CyberGreen, the qPCR machine will do what we call the melting curve. It's in order to verify the quality of the results. So it's a quality check, this melting curve. So the idea is very simple. So on the left, we've got, so in the y-axis, it's the RFU stand for relative fluorescence unit. So it's basically in the y-axis, it's the fluorescence of the samples. And we are at the end of the PCR, okay? And in the x-axis is the temperature. It's written here. So there are plenty of curves on the top of each other, but if you take only one curve, can you explain this? So we are double-stranded DNA. So all the CyberGreen is directly linked to the double-stranded DNA because the temperature is around 65. So we are in double-stranded DNA. So we have double-stranded, very highly fluorescent. If we increase the temperature, the DNA start to dehybridize. So there are the two-strand separation because of the increase of the temperature. I put the, so the more you heat, the more you denaturate DNA, because when you denaturate DNA, the cybergreen molecules will go away and the fluorescence is decreasing. And you arrive to a point, so this decrease is linear, and you arrive to a point where there is a big drop in the fluorescence in the tube. And that point is the point where all the molecules that were double-stranded, so all the PCR product double-stranded, are now in single-stranded, so completely denaturated. This curve, if you do, so it's the fluorescence relative to the temperature. If you derivate the fluorescence and you draw it relative to the temperature, so this is the derivate of this value. So here it's linear, it's equal to 1, so we are 1, okay, no problem. And here, this drop, so it's minus the derivate, so we've got a peak here, okay? And it's a very informative data in order to know if you have contamination during your PCR. So I explain why. When you are doing a qPCR reaction, you have using cybergreen. So you have two specific primers that should bind specifically to your target. So at the end, you should have only one PCR product, one bond. Okay. If you do a melting curve with a single PCR product in the tube, because there is only one type of molecule, you will have only one peak when you do this plot, this melting curve, because every peak is specific to the length of the PCR product and the GC content of the PCR product. If you add, for example, a contamination in your PCR during the reaction, which means that your two primers recognize two different targets, and in the same reaction, you amplified two different PCR products. These two different products, they will have either different size or different GC content. So anyway, they are different. And if you have two different PCR products, and if you analyze the melting curve, instead of having one peak, you will have two peaks. So the melting curve here, we use it to know if there are contamination during the reaction. At the end of a reaction, if you see a melting curve that is like that, linear at the beginning with a very nice and sharp peak, it means that your primer were very specific and you didn't amplify any non-specific target. But if you see two different peaks, very strong, it means that you get contamination. Did you understand this aspect? So it's based on the capacity of dehybridization of one double-stranded DNA with the increase of temperature. Okay, so as said before, we are going, so it's going to be quite short, is to give you the overview and the principle of RNA-seq. So, did you ever heard RNA-seq, the word RNA-seq?
I did hear this term before.
Okay, so what we saw before, so using qPCR or in situ hybridization, we monitor the expression of one single gene because we choose the gene and we monitor only one gene. RNA-seq is it's a high throughput analysis of the expression of all the gene in the genome at one time. Okay, so it's a global approach that is only possible with nowadays capacity of sequencing, because you will see sequencing is compulsory in order to do RNA-seq. The advantage, I think it's completely summarized in this slide. You've got two samples That's the minimum. You isolate the mRNA and you sequence. But you will not be able to sequence RNA. It's impossible to sequence RNA. So in this process, we extract the RNA, we convert them to cDNA, and we cut them in small pieces, and we sequence. So these are approaches in which you need huge amount of computing power to analyze all these things. The bioinformatic workflow are very complicated. And if you are going to do RNA-seq, I mean, you have to be full-time in that because it's a lot of bioinformatics and a lot of data analysis. But I explain you the principle because it's extremely powerful in terms of the result we can get. So with RNA-seq, you have to keep in mind that you are going to compare the whole genome expression profile coming from two different samples. So two different samples can be two different organs or two different points during the semantic experiment. So the idea is, what can you do? So I found this this slide. So it's a colleague of mine that gave it to me. He's working in medicine, but it's quite informative, this one. All what you can do with RNA-seq data. So we are not going to talk about all the applications that are explained on the right of the slide. I want to look at the A and B panel. So first of all, you have to see that part is done in wet lab and the other one bioinformatics dry lab. And this is why it's very complicated. Concerning the wet lab activities, everything is starting. We already saw that. So we extract, we take the tissue, so it can be tissue, culture cell, whatever you want. You extract the RNA and you do the cDNA synthesis. This is the very beginning and it's compulsory step. So you convert the RNA to cDNA because we can only sequence DNA. Then we use this. So when you see, for example, RNA sequencing, they don't sequence RNA. They sequence the cDNA that correspond to the RNA. Okay. So from all this cDNA that are coming from complete tissue, everything is sequenced, and we obtain only very short reads on the right and on the left of the sequence of the fragment. So technically is you sequence everything that correspond to pieces of cDNA and just short sequence, something like 50 base pair on every bit of it. So you produce a huge amount of data, and then it is all the bioinformatical process. So all the data will be cleaned, trimmed, and so on. And what I want to show you is what can be done with all this data. So we can, so this is all the process bioinformatically that can be done. But what you can do is, we can, from all this data, we can extract the differentially expressed genes. We call them DEGs, okay? So with this method, because we are going to compare two different situations, at the end, we will have two sets of genes with the number of occurrence of sequencing. So counting the number of occurrence in sequencing will give us the information about differently expressed gene. We can also, using that, identify new splicing pattern and alternative splicing. So there is this picture here, I think, that summarizes the most what can be done with RNA-seq. So you extract the RNA, you convert them to DNA, cDNA. You chop the cDNA in small pieces and you sequence the right and the left. So you end up with plenty of short RNA reads. So plenty of data like that. This is exactly what's coming out of the sequencer, plenty of small pieces. From that, there are two main roads depending on, are you working on an organism that for which the genome is completely sequenced or not. Okay, so if the genome of the organism you're working on is fully sequenced, the bioinformatical strategies are a little bit different. But anyway, we can have a lot of data in both of the situation. So we're going to see when we have the genome sequence of the biological model, what can be done. So with all these small reads, what we can do is align them one by one directly to the genome sequence. So we put them back in orders. Okay. When we put them back in orders, some bioinformatical routines, they will be able to give you, to reconstruct probabilities. Okay, so probabilities will be like that. For example, there is a color code here. So if you see there are purple, greens, and brownish boxes. The color code corresponds to the number of those sequences that have been generated. I explain myself. For example, this sequence has been found as many times as this one, as many times as the green, all the green boxes. The same for the purple one and the same for the brownish one. So from that, you can reconstruct what pieces goes with the other based on the comparison with the genomic sequence and the amount of redundancy between the sequences. And at the end, you will end up with something very interesting. So you assemble the transcript. You can find that for a single locus, so a single gene, for example, you have the brownish configuration, for example, that is always present at the same quantity in terms of number of reads, and that is always that contains always these reads. So this will show you that you've got one splicing alternative that is present, and you can find the other. And this is only due to the number of occurrences between the purple fragment and the brownish fragment. Do you understand a little bit or not? So basically, you sequence randomly all the ends of the cDNA that has been chopped in pieces. And from that, it's very complicated to explain because this is done bioinformatically. You count the number of occurrences of one sequence compared to the others. From that, you make groups of sequences that have the same number of occurrences, and this underlies the fact that all these sequences are coming from one single set of RNA, one single RNA. And from that, you can have conclusion saying, okay, this version of this gene, in the case of alternative splicing, is more abundant than this one, and so on. After, so this slide only show you one focus on one gene, but keep in mind that everything is genome-wide completely. So we can compare by counting the number of sequence read from one gene, the relative occurrence with the number of counts for another gene. And in that situation, when we want to compare the level of expression of two genes, it will be almost directly proportional to the number of the sequence, the reads for every gene. So it's a way that generates a huge amount of data, and it is the use of the number of identical reads that is going to be correlated with the relative expression of every gene compared to the others. Do you understand the concept? You link the number of reads to the level of expression of the gene. So I leave you imagine the amount of data. So it's true that after you process all this sequencing analysis, you can focus on splicing pattern if you want, but you can also look at for differentially expressed gene. You can do plenty of things. This slide is coming from a medical colleague, a guy which is working in medicine, and they are looking for variant in different genomes linked to genetic disease. You can analyze this kind of thing using RNA-seq. But for us, in general, we use RNA-seq to have the full picture of all the expression level of all the genes in one genome when we are comparing one situation to another situation. And this implies the sequencing of a lot of pieces of cDNA and the assembly and the count of occurrences. And everything like that to finish. And the results of RNA-seq, when we talk about DEGs, means differentially expressed genes. So we make we produce the result in that way because we can group the gene according to their behaviors. So for example, here, how do you have to understand? We will finish with this. Huh? This is the result of the analysis of the expression of, I think there is 500 genes on the left in a situation where, I don't remember the situation, but it's not a problem, after a stress, and you monitor that, so you extract the RNA half an hour after the stress, 1 hour, 2 hours, 6 hours, and 24 hours. And you have 5 different samples at the beginning. They did RNA-seq analysis, so they sequence all the cDNA in all the 5 samples. They sequence everything and they quantify the number of reads. With this data, you can group, you can make cluster of genes that have the same pattern of expression. The color code is here. If it's white, no difference in expression. If it's going to the blue, it's repression. And if it's going to the red, it's overaccumulation. So for example, we can let's take the first group of genes. So they are grouped because they've got the same expression profile, the same type. So this group on the top are genes that are very highly expressed at the beginning, half an hour after the stress. Then 1 hour, the expression decrease. 2 hours, almost nothing, and then it disappears. After, you've got other set of genes. If you look at that cluster here, that are not at all expressed and even repressed after the stress. And only 6 hours after the stress, the expression is increased. So you see how to read this kind of chart? So here we've got the time, the color correspond to the level of expression, the lines correspond to different genes, and they are grouped in the y-axis by similar pattern of expression over the time. You've got here this group of gene, all these genes are extremely induced very, very early after the stress, and then their expression completely drops down. Okay? Do you understand how to read this kind of data? Is it okay? Okay, so we're going to stop here for RNA-seq because you may have understood that there are plenty of different applications. I want you to keep in mind that it is a genome-wide approach that gives us, for us, the most important is that we have profile of expression globally at the genome level. So that's, and we represent the data like this heat map. That heat map. Okay, so we are going to make a break. If you have any question, you can tell me. And we're going to finish the lecture with the part of the RNA stability and post-transcriptional gene silencing. PowerPoint slides. So we're going to have a quick look to this, to another process. And we will end up with one, but I wanted to show you a little bit more technically how it works. So it's a process which is called, so there are different ways to call it. The general name is post-transcriptional because it's very precise, post-transcriptional gene expression regulation. So in other words, it's after the transcription process from the DNA polymerase, the mRNA are produced and they are not directly going to be translated into proteins, but there are very precise systems that are going to degrade the RNA in order to modulate the quantity of the RNA of a specific gene. So this is what, so I put the in full screen, this is what we call the post-transcriptional regulation. It's very complex. So the discovery of this process was completely by chance. It was first reported in the 90s, and the idea was really, the results were really strange. That's what stimulated the researcher. They were working on the pigmentation, so the color of the flower. So here, but the picture is not very nice, but this is the original picture of the paper in the plant cell. So the discovery was done in plants. It's really strange. So they were studying this flower because they wanted to change the color of the petals. But anyway, so the wild type flower is heavily colored in dark purple. It looks almost black, but actually it's purple. And the researcher, they wanted to increase or to change the content of the pigment inside the flower. So they did a transgenic plant that is overexpressing a gene. So it's not important to know. Anyway, it's called CHS. It's a gene that is implicated in the biosynthesis of the pigment. And they construct this gene under the control of a constitutive promoter. So basically what they did, they took a purple flower, a purple plant, they overexpressed this gene, so they were supposed to obtain more color in the petals. But what they obtained was the exact opposite phenotype. In other words, instead of spreading the color everywhere, they found some completely colorless flower, even if they were overexpressing the genes of the biosynthesis of the pigment. So they didn't understand at all what was happening, because instead of increasing the content of the pigment was completely disappearing. So later on, they managed to find the molecular, at least a link between the stability of the messenger RNA, so the direct product of transcription of the genes, and the presence of a new type of very small RNA that were 25 nucleotides long. So this experiment is very interesting. You see, it's just before 2000, and it's the first report, so it has been published in Science, of the identification of this new type of RNA, which are small antisense RNA. So I explain you a little bit this experiment. What they did, they used tomatoes for this study, and they wanted. So the idea is they were working on the fruit maturation because it's a very important economical problem. And the maturation of the fruit of tomato is under the control of a plant hormone, which is ethylene. Okay, nevertheless, so they wanted to modulate, using a transgenic approach, the response of tomato to this hormone. And for that, the strategy was to overexpress exactly the same situation that I showed you before, but it was the overexpression. So 35S is a constitutive promoter in plants that was expressing the ACO gene, which is a gene that is coding for an enzyme in the biosynthesis of this hormone. So I explain you, but it's not important to understand. So what they wanted to do is to overexpress this gene in tomato, and they generate different transgenic lines. So they are called T1.1.1, 1.2. So all those lines are independent transformant. So they are all transgenic plants that have received the transgene, but the transgene is located at different places in the genome because it's a random event. And they took these 5 plants, you see. So from these 5 plants, tomato plants, they extracted the RNA, and they wanted to do a northern blot to monitor the accumulation of the mRNA coding for ACU enzyme. Because they were overexpressing this gene, they were supposed to find very high level of the mRNA. Actually, this is what they found in 3 of the 5 out of the 5 lines. So you see T1.1 contain a lot of RNA coding for ICO. That makes sense. T1.2 the same, and 5.1 the same. But for some of the lines, and this is the case for lines T52 and T53, even though they were overexpressing the ACO gene, no mRNA was seen. So in this context, all the RNA that were overproduced by this construct was directly destroyed and degraded. And at the same time, when they did this experiment, to be sincere, it's a little bit by chance. They monitored as well population of RNA, because it was in the same gel, okay? Population of RNA, but very small in size. And they realized that when you have high amount of accumulation of the native mRNA, you don't have small RNA corresponding to this RNA. But every time you see the disappearance of the RNA that you would have liked to overexpress, it was concomitant to the appearance of small RNA of 25 nucleotide long. So they make a link that the disappearance of the full-length messenger was correlated with the accumulation of small RNA that are the same sequence. So this was the very beginning of the discovery of this process. So I summarize a little bit what has been done till this time, because a lot of years passed by. From now, there are plenty. It would take hours and hours to talk to you about all the different microRNA. But basically, there are plenty of different types of small RNA. So I gave you example of the size. They can be 21 nucleotide long, 20 to 24, 26 in some cases. And they have been discovered in plenty of very fundamental processes. And I will show you two of them just after showing you the mechanism. So they are now divided in 3 main classes. I would say 3 main classes. There can be others. They are the one that we call the microRNA. You heard about microRNA before or not? Okay. So the microRNA, they have the characteristic to be 21 nucleotide long, and these microRNA are coded by specific genes that codes only for microRNA. It means it's a gene that is producing an RNA after transcription by POL2, and the RNA is never translated. I will show you how it works just after, and it just gives rise to this small RNA of 21 nucleotide long. Important, and we will come back on that at the end, this gene, the gene coding from this microRNA are transcribed by RNA polymerase II. Okay. So these are, let's say, proper genes. Okay. Second class, very different because they are not coming. They are not. Their origin is not the same of the first class, the microRNA. They are all called in this group small interfering RNA, and the name that you probably have seen is siRNA. Their size can span from 21 to 24 nucleotides, but their origins is very different from the microRNA, even though some of the RNA pool can be the same. MicroRNA are coming from dedicated genes, whereas small interfering RNA are coming from secondary structure from RNA, like hairpin. So it's a loop in the RNA that can be recognized. Some of the genes that are producing these strings are encoded, are transcribed by POL2. They can come also from specific structure in the genome, which is called natural antisense RNA. It's not very important, but you have situation in the genome where you have two genes that are overlapping one each other, and one is transcribed in one way, the other one is transcribed in the other way, and the overlapping part is going to give rise to double-stranded RNA that can be converted in small RNA, but anyway, it's not a problem. And there is also some of the sRNA, the one that are 24 nucleotide long, that come from some heterochromatin structure. And this is quite complicated because there is a link there between the chromatin structure and the presence of this small RNA. So you understand why the world of small RNA is very complicated. After, there is a third class that is not present in plants, specific on animals, that are the PIWI interacting RNA. There are 26. And PIWI is implicated in different processes in stress. Anyway, we cannot make the list of this small RNA. It was just to show you that there are different classes because these groups are made based on their size, but also on their origins. Okay, so some of them are derived from secondary structure of RNA. Others are direct proper genes, and the way they work is extremely complicated. So you don't have to remember that because you will see it very more in depth next year. So this is a kind of summary. We're going just to spend a little bit of time on that, on what's happening, and I will try to show you what is important is to see the identity of this pathway. So vertically, you've got four processes that can, if you look at the bottom of this table, this is what they are going to do at the end. So 3 pathways here. The first 3 one from the left. The consequence of this small RNA, I will explain you after how it works. The presence of this small RNA has as a consequence the degradation of messenger RNA. So what you have to keep in mind is that these small RNA, they will at one moment manage to participate to the degradation of target's mRNA. And therefore, it will modulate the quantity of the RNA. I'll show you something after. So for the microRNA genes, it is very simple. The strategy, what is a microRNA? It's a piece of DNA contained in genomic DNA that contains a promoter, a full promoter, that is transcribed by RNA pol 2, as it was written in the previous slide. When this gene is transcribed, it's going to form an RNA that is a proper RNA coming from the transcription of pol 2. So it means that there is a poly A tail and so on and so on. But this RNA has a reverse and complementary sequence in one part of a sequence, and it forms a kind of stem loop secondary structure. This type of RNA is never translated and will never give rise to any protein. From this miRNA precursor, it's then charged to a complex, cut. There is only one piece that is going to be maintained, and this is what is called the miRNA duplex. This is the proper microRNA. And this microRNA is going to be taken by a complex of protein called AGO for Argonaute with the RNA inside that is going to specifically target the identical RNA complementary to the sequence and degrade this RNA. Do you understand the process? It's important that you understand this process. The other one, they are very, very similar. So this gene is producing a precursor of a small RNA that is going, this small RNA will target specifically this complex to a messenger RNA complementary to this small RNA, and the consequence and the action of this complex is to degrade, to cleave all the target RNA. So you can say as a conclusion, when a miR gene, and I will show you a very nice example of this, when a miR gene is active, it will destroy the mRNA that are produced by another gene. So it's a way to regulate the steady state level of RNA, not modifying the transcription level, but modifying the mRNA degradation. Okay. After there are, as I told you, the origin of the small RNA can be different. So there was what we call TAS gene is transactivating gene or cis antisense genes. This is the genes that are in front of each other, like we see. At the end, you see the process is extremely more complex anyway, but at the end, it's always the same. From an information that is coming either from a gene or from an external exogenous RNA, a small RNA is produced, and this will serve as a guide to direct the degradation of another mRNA target. Okay, so this is the three main pathways. Do you understand the concept of this pathway? You don't have to remember all the name of the proteins involved in that, because it's always growing and it's quite complicated, and you will see that next year. But do you understand what I explained you on the mode of action of microRNA gene, for example? Is it okay for everybody? So consider that the three pathways on the left, they are very similar, not in the way they are working, but the consequence is degradation of RNA. I just want to highlight the fourth pathway, the one that is presented completely on the right, that is completely different, but in the center of that, you have these small RNA that are playing a crucial role. So I'm just explaining you this, and after we won't never talk back again, but it's very important. I will explain you where they are coming from after. So you have some small RNA that can be produced. They will be charged in this same type of complex, degradation complex, okay? But instead of being implicated in mRNA degradation, this specific complex is going to be the RNA sequence of the small RNA is going to target this complex to the genomic DNA, and the consequence of this genomic DNA recognition will be DNA methylation and chromatin condensation. Okay. So you see here, it's becoming to be very, very complex. We are talking about small RNA, and the mode of action of this small RNA is going to target specifically some part of the genome in order to process DNA methylation and chromatin remodeling. So where these small RNA are coming from? These small RNA, they can come from, I told you when we talk about the structure of chromatin, that the condensed chromatin, under the condensed chromatin, there is very, very often, most of the time, repeated sequences, and there was a lot of transposon and retrotransposon. And for the cell, it was very important to block the possibility of transposon to jump from one place to another place. What is happening is that this process that is on the right of this slide is the mechanism of the detection. So these small RNA, which are called HCSI RNA, are produced when some of the repeated sequence and transposon are jumping. So the cell can recognize the activity of some transposon or viruses or endogenous viruses and directly target the condensation of chromatin at the place where they jump from. Okay, so it's another completely different mechanism, but it was to show you that, okay, those small RNA, they play a major role in the control of RNA stability because they actively participate to mRNA degradation, but they can also serve as a guide to modify the methylation state of DNA and therefore the chromatin condensation state. Okay, so I'm just going to show you one example of this type of regulation because it will make more sense to you. So okay, you know, this is okay, right? This is easier. It's a zoom of this type of mechanism, so you can see the microRNA pathway and the small interferon pathway. It's exactly what is presented in the previous slide, but it's a little more detailed. And I just put this slide also because I wanted to put you this this HYL1 in the biosynthesis pathway because we're going to show a mutant of this. So I'm going to show you. So this microRNA pathway, I'm going to show you a specific case. So just to be sure. So this is the primary transcript of a microRNA gene and all the process that gives rise to mRNA cleavage or RNA translational repression is not a problem. The first partners that are necessary in order to process the pre-mir RNA are a complex, and HIL1 is one of these complex. And I want to show you this this experiment. So just to show you that what we saw, the microRNA regulation of the steady state of mRNA level, it's completely necessary for development. So here it's a plant. So this is Arabidopsis thaliana. On the left, you've got it wild type. It is at the end of its life because it's flowering. Okay, so this is the wild type plant. And this is the same plant, but mutated in HIL1. So you see the phenotype, it's completely different. So imagine this plant is not stressed at all. It's just normal development and the development of a mutant. I just go back. The only difference between the wild type plant and the mutant that you just saw is this gene is not active. So imagine that all the MIR pathway is completely implicated in the development of the plant, of the normal development of the plant. And it's the same for animals. Then this is also the result that show that the small RNA are also implicated in the response of plants, for example, or other organism to external signal. So here it is the phenotype of a wild type plant or the same mutant, this HIL1 mutant, to the application of plant hormone. Don't worry if you don't know this plant hormone, it's not a problem, but you see that a wild type plant, when you treat it with this ABA molecule, the phenotype, it's a normal phenotype. It loses the green part, it's turned yellow, and the development is reduced. So this is the normal phenotype. If you check the same phenotype in a HIL1 mutant background, you see the phenotype is even more severe. Okay, so the conclusion of this slide is that this mutation HIL1 is only affecting the microRNA pathway in normal development, and you see that if you affect fundamentally the production of microRNA, you affect normal development, and you affect also the response to external stimuli. Okay, so it's a process that is completely normal and natural. Here you have, a molecular evidence on how the microRNA are working. So we're going to analyze this result. So on the left, this is northern analysis. So we measure the quantity of specific RNA. On the left, we measure the accumulation of the microRNA. So they are named like that. They've got numbers. So we are monitoring the accumulation of 3 microRNA. 159, 167, and 171. Okay, so this is the profile of this microRNA in different genetic background. I will explain you which one, which genetic background it is. On the right of the slide, this is the same northern analysis, so quantity accumulation of messenger RNA of the target of this microRNA. So, for example, the microRNA 159, if it is produced, it's going to recognize the MIB33 RNA and degrade this target. The MIR167 is going to recognize and to degrade ARF8 target. And the 171, it is this target. Okay, so we're not going to analyze everything, but look what's happened. Let's take the wild type lane in both situations. Do you see the relation between the accumulation of the target and the accumulation of the microRNA? Do you see a relation? When the microRNA is expressed, it will bind to the target and the target will be degraded. So when the signal of the microRNA is high, the signal of the target is low. Okay. Then if you check the accumulation of these microRNA in different mutant of the biosynthetic pathway of the microRNA, like HIL1 that we saw. In HIL1 mutant, the microRNA, they cannot be produced. I showed you before. Oh, sorry, is this one? If HIL1 is mutated or HEN1 as well, the microRNA here, they cannot be produced because this step is blocked. So in the mutant HIL1 and HEN1, the microRNA, they cannot be produced because of the mutation. So you see they are produced in the wild type, but not in the mutant. And the consequence of the absence of this microRNA is that the target now is not degraded anymore. You see? Do you understand this now? It's fine. And if you, of course, if you overexpress these genes, it will be the same as the wild type. Okay. So you see, what I wanted to show you is that it's a way, so it means that the control of expression of this microRNA can control the steady state level of other mRNA, which are the target of this mRNA. So I will show you, because we're going to finish. I don't want, so it's don't take care about the don't pay attention to this one. So one example, and I hope it will be a little bit more clear on that. So an example of how the microRNA can control, can affect the genetic development. So that's not the case of a plant, but it's the same. So there is a specificity of the leaves of plants, of dicot plants, is that the cell types that are on the top, the upper face of the leaf, are different from the cell type which are underneath the leaf. So the top of the leaf, it's called the adaxial face is the name, and the bottom of the leaf, it's called the abaxial side of the leaf. And the two different cell type that we found is just to show you the system at the beginning. So on the adaxial, we've got trichomes. Trichomes, they are little hairs on the surface. And on the abaxial face, we've got the stomates. And the stomates, these are this part that are implicated in respiration of the plant. But anyway, so it means that a leaf of a plant has a different cell layer on the top than the one from the bottom. So two different cell type. Okay. So this is the starting point of the example. So there have been plenty of research that has been done in what is the genetic control for the cell fate on the top compared to the bottom one. So the researcher at the beginning, they were thinking that there were specific genes that were expressed on the upper part of the leaf and specific genes that were expressed in the lower part of the leaf. So that makes the difference between the top and the bottom layer of the leaves. But after a lot of research, they found out that everything was under this type of regulation. So I explain you the way this schematic is done. The left part of the schematic is what is the situation in terms of regulation in the embryonic state of the leaf. It means before there is differentiation between the abaxial and the adaxial. So it's the state of the system before differentiation. On the right of the leaf, of the slide, it's what are the situation in terms of gene regulation in the adaxial cells, so the one on the top and the one on the abaxial side of the leaf. So everything, so we have to look at this. Do you understand the way it's presented? So it's before the specification, and then once the two different sides are specified with their differences. So let's imagine one thing is that, so you see we've got one gene, so the code is like that. The boxes are the genomic DNA, so one gene, this is the transcript, and the small balls are the proteins, okay? So what we have to look at this when it's written response gene. So it means that this is the gene. We don't mind. We don't care about which are the genes for this. So these are the response genes that are going to be transcribed and translated. And once they are translated, you will have. So when these proteins are translated, it means that the cell type will be the adaxial leaf fate. Okay, if you have a cell that does not express this red gene, you will be in a situation of the abaxial leaf fate. Do you understand what I mean there? It means that if a cell type contains the red protein, it's going to give rise to trichomes, so on the top. And if the cells doesn't have this red protein, they're going to form stomates, and it's going to be the bottom of the leaves, okay? So in other words, when you have the red proteins expressed, it's the top of the leaf. When they are not expressed, it's the bottom of the leaf. And the mechanism is working the regulation to know which cells are going to express the red protein and which are not going to express is under the control of a microRNA gene. And the regulation is like that. So you've got a master regulator. It can be a transcription. It is a transcription factor. It's called PHV. At the beginning, before differentiation in different cell type, this transcription factor is transcribed and translated. So the protein of this transcription factor is present. This transcription factor can recognize the promoter of the response genes, okay? And it recognizes the promoter of the response genes, which are going to be transcribed and then translated. So at the very beginning of the developmental process, all cells are in that state. Then, once there are external signal for developmental progression, there is another gene, which is a MIR gene, that is called MIR165-166, so that's his name. This gene is going to be expressed in some cells, so it's going to give rise to the microRNA, so the 21 nucleotide long. This microRNA is going to recognize the messenger RNA of PHV-PHB, and therefore this PHV-PHB is going to be degraded. Because it is degraded, there is no protein PHV produced, and the response gene won't be activated. And so you will say to me, okay, the system is like that. So at the beginning, before differentiation, all the cells express the response genes. So they should all turn to be adaxial leaf fate. But only in the cells that are going to be the abaxial cell layer, only in that layer, this gene, this MIR gene is expressed, and the consequence of its expression is the degradation of the regulator, and so you don't have any more expression of the target response genes. So you see the process here. Do you follow this process? Can you tell me? It's okay. I'm just going, and then after we finish, I'm just going to no, it is to show you one thing. Anyway, we have Okay, so just to show you because even if you understood, if you have, so this is, let's say no, how am I going to do it? We could have imagined one thing. So I draw the same promoter. Okay, and I can say, so this is abaxial, and this is addaxial. Okay, so this is the response gene. So you see, in the example I showed you, the response gene were active only in the abaxial cell of. Really, you have one transcription factor that is present only in the abaxial cells. So exactly the same as the growth hormone factor. So if this transcription factor is only expressed in this abaxial cell, it's going to be able to activate the response gene. But in that case, so the gene, how can I say, the expression of the transcription factor is expressed only in abaxial. But in the case, in the situation that I showed you, the transcription factor, the messenger, okay, I will write it like that. The messenger of the transcription factor are produced in both of the cell type, but the microRNA destroy the RNA only in the abaxial cell fate. Okay, so it's a way. Sorry, I need to reopen. Okay, sorry, I just share it back again. Okay. So you see the biggest difference from what I tried to draw you in that situation. So this is the transcription factor. And there was one way of arriving to the same process, which is, for example, if this transcription factor was transcribed and translated in the addaxial cell, the transcription factor is produced and is going to activate the gene, so you have the response, no problem. But in what is very strange in this situation is that in the abaxial cells, where the response genes are not activated, the transcription factor is transcribed, but it's never translated. It is the mRNA is degraded directly after transcription. So if you compare to the exercise that we did on the growth hormone factor, we saw that, for example, the transcription factor, so the equivalent of this one, of PHV, so the transcription factor GHF, was expressed in the anterior part of the pituitary, and it was because it was expressed and translated that the GAS gene was active. In that case, it's different because the transcription factor, which is PHV, it is transcribed in the abaxial cell, but it cannot be translated because of the degradation that is stimulated by this small RNA. So then, because the protein is not there, it can never activate the other genes. So it was to show you that what conclusion can you draw from that is that you see that in this drawing, there is the two processes at the same time. There is the transcriptional activation, the activation of transcription by transcription factor. So it can give rise to production of messenger RNA and then protein. But at the same time, the transcription of some microRNA genes will have as a consequence the degradation of specific messenger RNA. So we have to think globally that the accumulation of mRNA is the result of the production, so the transcription of this mRNA, and the degradation. So you have to consider transcription plus degradation to know if the RNA is present or not. Is it okay or not? Okay, so do you have any question on that? After, you will see that in really much more detail on specific point, especially when you will do some lecture about some pathogens and some viruses, because one of the way for eukaryotic cells to fight, for example, against viruses, is to directly degrade the RNA that is produced by the virus. And this degradation implicates the same process. So it's specific recognition of viral RNA and degradation of viral RNA. But you will see that in other teaching units. So to end up, I have just 3 very simple slides to make a kind of summary, because I talked to you about this central dogma of molecular biology. So it was just to re-put a little bit all the concepts that we saw during this course all together. So just to tell you this central dogma, as one of you told me at the very beginning of the lecture, it concerns the flow of information that is stored in the nucleic acids. So it's the concept rose in the 60s. The idea is brilliant, it's perfectly true, but it was completely, at that time it was perfect, but it's not very, very complete. So if we put all what we saw together in this model, we will end up with something a little bit more complicated. So just a quick think about information flow. When I'm talking about information, is data stored in nucleic acid and or proteins as well. So DNA information is multiplied on DNA. This is replication. Then the information from DNA to RNA, this is transcription. RNA to protein, this is translation. And we have all this process. So replication, transcription, and we saw this. We saw transcription, RNA processing, RNA from protein translation. Then you know about post-translational modification. So here, these are all the partner or the concept. So this is very, very global, and all the red points implicates information transfer. If we put all together what we saw in this module, we can add more relation between objects. So I will try to just describe you what we saw and try to re-put in the context of this central dogma. So, for example, the first flow of information from DNA to DNA, it is replication. But you remember that we saw that there is another type of information that can be analyzed and transferred, was the DNA methylation and the chromatin structure. Heterochromatin structure is maintained over cell division. So we can add one more arrow. After, it's written everywhere. We saw also very quickly that, for example, the information that is stored in RNA can act on DNA. we saw it very quickly, but when I said to you that some small RNA can be the guide of de novo DNA methylation or chromatin remodeling, it means that they are implicated in information flow from RNA to DNA. All what we saw, for example, for the microRNA, the classical microRNA we just saw a few minutes ago, these are RNA, small, okay, but these are RNA that contains an information, and this information is going to be directly perceived by RNA because they are going to be degraded. So there is also information from RNA that is going to affect RNA itself. After, we also saw for this flow of information from RNA to RNA, we also saw the alternative splicing. And if you remember the alternative splicing, so you change which intron you remove, but the removal of the intron, they are done by complexes. It was U1 and U2. And these complexes, they contain protein and RNA. So it means that to splice RNA, you need RNA. So they are linked together. After, there is another part that we saw very much in depth is the information stored in the protein. How can they affect DNA? We saw chromatin remodeling processes. So this protein, they can remodel the chromatin that has, as a consequence, DNA methylation, and so on and so on. And the proteins are part also of the information flow toward RNA through editing and splicing, like RNA on RNA, and so on and so on. So you see the concept, what I wanted to show you is all the little bricks that we saw all together during this few lectures. We can link them all together, and you see that the model, the original model is this one, is completely right, and it was very, very clever to propose it. And now we have much more interaction between partners that we thought at the beginning that they were downstream this process, but they are acting on the top of the process as well. Okay. So this is what I wanted to tell you. So I don't know if you have any question in that. Do you understand just all these new arrows on this model with all the things that we saw during this this module?
Can you repeat the part about the prions?
So the prions, we never talked about that, but do you know what is? They have been found in diseases. You remember this mad cow disease and so on? Okay, and basically it's a protein, okay, that can be folded in two different way, okay. Normally the protein has to be folded in one way, okay, and for one reason, I don't know which one exactly. If it is folded wrongly, okay, the consequence is that this wrong folding is going to be spread all over the other proteins. So it's a bad folding of one of the protein is going to affect the folding of all the other one. This is why I put a loop in terms of information, because it is the information stored in the secondary structure that is transferred to the other secondary structure. We didn't see it because it's the protein world.
Okay.
But you understood what I mean? So you can check. There are some summary on the internet, but technically it's a protein. If it change of conformation, it will affect the conformation of the other normally fold protein. And so it's a self-amplifying phenomenon that gives rise to disease. But do you see all the arrows? For example, if I ask you, it won't be the exam, but an exam, if I draw this with only the arrows, can you give example for every arrow?
Maybe.
For example, if I ask you, give me an example of the effect of protein on DNA. But you know that the protein, they can affect the condensation of protein, of the chromatin, and the condensation of chromatin can affect methylation of DNA. So what I would like you to do is to try to put back all the bits all together using this drawing. For example, for the influence on RNA on itself, the small RNA have an influence on the other RNA because they degrade them. And the RNA present in the spliceosome, they are important for splicing and so on and so on. And we saw plenty, except the prions, we saw always example of all these arrows. It's okay or not?
Yes, it's okay.
Okay, so we have finished. So I don't know if you have question. Anyway, if you have a general question altogether, if you want that I give you more detail on something, you can still discuss all together and send me your question in one document if you want. If you want that, if you want to have more precision on specific things. Okay. Anyway, I would like to thank you for being present during these 15 days, and I'm really sorry of having to give the lecture by Zoom because it's very difficult. Normally, okay, we don't know each other, but normally when I'm doing lecture, I need to have a real blackboard, and I'm using a lot of this to that. But doing Zoom, it's very complicated and it's not well adapted to this kind of lecture. Anyway, I hope that that you learned some things with this, and I'm pretty sure we're going to see physically each other in spring because I will be in Hanoi starting from end of April or something like that. So I hope we can meet all together. Okay. So thank you very much for your attention, and please do not hesitate to contact me if you want to have more details on some point, because okay, if you want any information or more explanation on specific things. Okay, so thank you very much for your attention, and good luck for the next lectures.
Okay, for how the final exam?
For the final exam, we don't know yet. We are trying to find a date with Hang, but it should be, as we said the day before yesterday, it should be second half of February, something like that.
Okay, thank you.
Okay, you will be informed really before, don't worry. Okay, so have a good end of day, and hope to see you soon. Bye.